Hi everyone. So we're going to have another important webinar today, and this webinar is based on the Unit 2, Topic 3C, the organism growth and development. So this includes cell differentiation, interaction between genes and the environment, controlling gene expression, stem cells, and using stem cells. So I have distributed already the questions. Now we're going to find a solution and explanation for those questions. Right, we start with the first questions. Linked genes are usually inherited together. The diagram shows the position of two genes on three chromosomes, chromosome A, chromosome B, and chromosome C. So chromosome A, chromosome B, and chromosome C. And we can see gene 1 and gene 2 here. This is the 2 gene. And here, this is the 2 genes. And here, this is the 2 gene. Name the part of chromosome that is occupied by the gene. So when we are trying to locate the gene, this place is called locus. So gene 1 and gene 2 location is also called locus here. And what we can see, the difference of this gene 1 and gene 2, they are very close together here. And they are apart from here and then further apart from chromosome 1 and 2. Explain which chromosome shows the weakest linkage between gene 1 and 2. So the linkage is stronger when they are close to each other. The linkage is stronger when they are close to each other. For example, this is the chromosome and they are located very close to each other. This has a strong linkage. So the weakest linkage is weaker when they are apart from each other. From here, this is located here and this is located here. So linkage is weaker when they are located apart from each other. So these are the linked genes. They are located on the same chromosome and they are close to each other. This is a linked gene, and these are not linked gene. Okay, so if you look at this gene 1 and gene 2, we can still come up with solution. So go back to the previous one. Now, during meiosis, we know that during meiosis, then chromosomes are overlapped. For example, homologous chromosome for this one overlap here and overlap here, right? So we assume this is the overlapping position, and after that we know the part of the DNA chromosome exchange. So this is called chiasma. The location is called chiasma. For example, this one is a chiasma and this one is chiasma for this chromosome. Now it's overlapped. After crossing over, then they are separated. After separation, we can see the blue one which carries the other one, and this part is coming from black one. So black chromosome is this one, and this one is coming from black, and the black one So I use the black color then. So black color, and then I use blue one for this. This is the blue one. So black color get the blue one, blue color get the black one, and that's the exchange of the DNA. Now this gene, which is located here, now moving to this place. So then still there is a chance separation of the gene when they are located on the same chromosome. There is a chance to separate the genes when they are located on the same chromosome because of the crossing over. But unfortunately, if they are very close to each other, for example here, now you can see this is the crossing over place. When they cross over and then exchange their chromosome, part of the chromosomes, you can see this get a new piece from the overlap one, and the gene 1 and gene 2 is located to the other one. So when they are very close to each other, there is a very less chance of separation. So then crossing over, it can't. So then if we go to the genes are located separately apart from each other, so gene 1 and gene 2, and crossing over is here. After crossing over, this gene 2 is going to the other chromosome, and gene 1 stay in the old chromosome. So same thing with the C. So they are very distal to each other. When they cross over, you can see gene 2 move to the other one and gene 1 stay in the old chromosome. So that's the story of this link gene. They are very close to each other, and during crossing over, they still inherit together. So answer for this question, A, B and C. So weakest linkage we can find C, and strongest linkage we can find in A. So I write answer chromosome C, which has the weakest linkage, because the genes are furthest apart, more likely to be separated during crossing over. Chiasmata forming between the two genes. So chiasmata form and they separate, and the chromosome C has the weakest linkage. Next question, B part. The process of meiosis give rise to genetic variation. So give rise to genetic variation mean the crossing over and random assortment provide the variation. Gene A, B, C are located on two different pairs of chromosomes as shown in the diagram. So this is a homologous pair. It has one gene, gene A, and the recessive one, simple A. This is a homologous pair, and it has a B and C. So if you look at B and C, they are on the same chromosome, and they are not very close to each other. We can consider these are not linked gene because they have some distance. When they have a distance, there is a higher chance of separation. So this is simple B and simple C. Now these are the two homologous chromosomes. During which combination allele could be present if crossing over has occurred? So meiosis gives rise to genetic variation. Gene A, B, C are located on two different pairs of chromosomes as shown in the diagram. Now what will happen? After the crossing over, they separate. For example, gamete formation. So you can understand this one, effect diploid here, and this is a diploid. Only one chromosome is coming to the gametes. One is here, and the other one is here. So there are various combinations we can write. For example, the one combination, looking at this one, I can say if this one is coming and this stay here, and this one is coming and it has B and C. So we have A, B and C. But question is asking what will happen during meiosis and crossing over take place. So crossing over take place, homologous chromosomes, then they overlap. So there is this one has no link gene. It's only one gene. Only crossing over take place here to exchange the part of the chromosome. I hope you understand. After that, we can find what is the best combination and most possible combination we can get. Now, let's say this is a one gamete and this is a second gamete. So it's quite clear this one has only one gene, recessive one and dominant one, and they clearly separate to two gametes. Now we have the homologous chromosome with the linked gene. They are distal to each other. After overlapping, they exchange part of their DNA. So here it could happen like this. Now, if they cross over, simple B stay as usual and capital B stay as usual. But here, this part, the crossing over take place, distal end, this end crossing over take place, and it swap. This capital C goes here, simple C goes here. Now this has simple B and capital C, other one has capital B and simple C. So then I can expect simple A and this goes to the gamete simple B and capital C, and this one stay here and this goes to the gamete, and we can get capital A, capital B and capital C, because capital B and C is coming from the other chromosome. So then answer is capital A, capital B and simple C. So as you see, the crossing over and gamete formations are very important event to bring the variation. You are frequently checked this knowledge in the exam. Right, question 2. Question 2 is based on a sex-linked gene. You can see X chromosome and Y chromosome. Hemophilia is a condition that results in excessive bleeding due to lack of blood clotting factors such as factor 8. It is an X-linked recessive condition, and one cause is a mutation to the F8 gene. So let's say there is a gene called F8 on X chromosome. If there's a mutation, it cannot produce correct proteins and it affects blood clotting. Complete the diagram by drawing a line to show possible locus for F8 gene. So F8 gene is located on the X chromosome. So you have to clearly show it on the X chromosome. Now you can draw a line like this to show F8 chromosome. There is actually a right locus for F8, but you are not checking where is exactly located. So you can write it anywhere on the X chromosomes, this F8 gene. Explain with the use of genetic diagram why males with hemophilia cannot pass on the condition to the male offspring. Right now, this is a father and this is the son. The father has XY combination, son has XY combination. You know, the Y chromosome, actually this diagram is not drawn to the proportion. Usually the X chromosome is very long and Y chromosome is very short. As a result, X chromosome also carries genes which is not only for sex determination but for other phenotypes. Now, father can has a gene on X chromosome, and son also get gene from X chromosome. So what will happen during gamete formation? This X and Y. Now you see a father has X chromosome and Y chromosome. Now when you get a mother side has X and X. Now you look to make a male, right? So you need definitely Y and Y. But X is coming from mother. X is coming from mother. And if you look at the other one, this X is coming from father, this X is coming from father, and this sex is coming from mother. So this is a male and this is female. Now what you can observe, the always Y chromosome goes to male side and male X chromosome is coming from female side. Explain with the use of genetic diagram why males with hemophilia cannot pass this condition to the male offspring. So let's say the red dot indicate the hemophilia, the mutated allele, let's say simple H. So simple H always goes to female side. It doesn't go to male side. So I further showed this in this genetic diagram. So this is a complete genetic diagram. So this is a hemophilia X simple H, and male can express the condition with single allele because Y doesn't carry any alleles. So that's this one belongs to the father and this belongs to mother. So mother is completely normal. Mother has no hemophilia. That's a homozygous dominant, capital H, and this is represent mother's and father combination. So this is female, this is coming from father. This is female and the mutated allele is coming from father. When you get the son, completely normal, only Y is coming from father and X is healthy one is coming from mother. So male parents pass their Y chromosome to the male children. Remember, male parents pass their Y chromosome to the male children. Male parents can only pass their defective alleles to the female offspring. That means X chromosome pass only to the female. That's a main idea. So indication of position only in X chromosome, that's a line. And then 2 marks. When you highlight these 2 with the genetic diagram, you get 4 marks, right? So genetic diagram 1 mark and showing X chromosome 1 mark, and giving these 2 points, you get a 4 marks. This is a very strong one, better to remember. So male, the father can pass Y chromosome only to the male and father can pass X chromosome only to the female. Just a rule. Okay, next question. There are 2,931 mutations to the F8 gene that have been identified. Now, you know, there are many mutations we can find in genes. For example, cystic fibrosis. Again, it has many mutations. So likewise, we cannot always get one mutation, but some condition like sickle cell anemia, only one mutation. Hemophilia can also cause by mutation to the F9 gene. So F8 and F9 gene, 2 genes. So this is F8 and that's F9. So hemophilia come from mutation of any F8 or F9 gene. Hemophilia is not classed as a polygenic disorder. Give the meaning of the term polygenic. Now poly means many genes. So it is not related to interaction of many genes. It's very clear if there's a mutation of F8 and that's directly affect to the proteins. It's not the combination of many genes and outcome or phenotypes. So polygenic is usually continuous variation, and polygenic mostly we can see, for example, skin color, more than 150 genes are known to affect the color. That means polygenic means the genes are located in different chromosome, different locus, and their interaction can cause the phenotype. So definition is a phenotype that displays a range of variation is influenced by several genes located at various positions. That's a definition of polygenic. Now this calculation, there are 2,931 mutations to F8 gene that have been identified. Hemophilia can also cause by F9 gene, which has 1,133 mutations. Now look at here, there are 575 identification So hemophilia can also be caused by mutation to the F9 gene. There are 1133 mutations to F9 gene. There are 575 identified mutations to F9 gene that cause severe symptoms of hemophilia. That means severe. Others are mild. So total mutations are this much, and 575 mutation can cause severe hemophilia. Then what is the percentage? So we can calculate the percentage. The same percentage of this type of mutation is found on F8 gene. So F8 gene, the percentage is same. So let's calculate the percentage. Now 575 divided by 1133, we get 51%. So F9 gene, 59% cause severe hemophilia. So that's the same here, 51%. So then they are asking calculate number of mutation. That means X we don't know, and total number is 2931 and equal to 51%. So get the value of X. So we can get the value of X by multiplying 2931 by 0.1%, 1495. So this is the solution for these calculations. Right now, we'll continue the the next question is based on stem cell. So we have learned stem cell, and the definition of stem cell is these cells are very special cells. So these cells can divide endlessly. So if we have an embryonic stem cell, we can say these stem cells are the best stem cells, and there is no limitation, and usually we call they can divide endlessly. But when you talk about some adult stem cells, maybe there are limitations. The second thing is, so they can develop or differentiate into many different type of cells. I mean, any kind of cells. If you take embryonic stem cells, theoretically, they can differentiate into any kind of cells in the body. The last thing is, they are undifferentiated cells. So they can self-renewable and undifferentiated cells. These are the three criteria of the stem cells. Now, when you look at the stem cells, we have limitations. That means embryonic stem cells are very difficult to use in experiments because of ethical issues. So usually animal stem cells we can use to understand some of the mechanism of stem cells, and also we can use adult stem cells to understand and also therapy for some of the conditions. So what we can do, we take the stem cells and then we can differentiate into many type of differentiated cells. So in this question, what they have done, they're going to use some of stem cells from organ. That means stem cells are found in some organs and tissues, including the brain, bone marrow, blood vessels, skin, teeth, and heart. So scientists are continuously trying to discover stem cells in these tissues, and they're trying to differentiate these cells into the tissues where they're isolated. For example, this question, stem cell can be used to repair damaged organ. That's one medical use. We can use stem cell to repair the damaged organs. Stem cells from the heart can be used to produce mature heart cells. So stem cell from the heart, we can use heart mature cells. The diagram below shows one method used to produce mature heart cells. stem cells from the heart. So they isolated stem cell from the heart, and after that, what they have done, they have used the whole heart chemicals isolated from the heart tissues. So we know that chemical stimulus is important, and for that they have used the chemicals from the isolated heart, chemicals isolated from the heart tissues. And after that, when they add to immature heart cells, so they become a mature heart cells. So stem cells isolated, and they allow them to make immature heart cells, and using the chemicals they make the mature heart cell. The whole heart contains cardiac muscle tissue. The whole heart is an organ. Give one difference between tissue and organ. So that's a theory part. Tissue is a similar kind of cells working together for a function or functions. And organ is collection of several tissues working together for a function or functions. So group of cells with simple similar structures working together. I think it's important to write this word working together to perform a shared function or function. So this is a group of tissues again working together is important word to use to perform specific functions. Right. This is the answer. Tissue is made of one type of cell, and an organ is made up of different tissues. That's the difference between organ and tissue. The next question, part of the eye contains stem cell. Okay, so eye also has a stem cell. These cells can be used to repair damaged corneas in the eye. Explain why stem cells from the heart cannot be used to grow cells to repair the cornea. Why stem cells from the heart cannot be used to grow cells to repair the cornea. Can you remember the three types of stem cells? We have learned three types of stem cells. So these stem cells basically we can divide into two. One is stem cells, two types coming from embryos, and one type is coming from the adults. So adult stem cells and embryonic stem cells. So adult stem cells, two type, totipotent, that means they can give rise to any cells. Pluripotent, meaning they can give to any except extra embryonic tissues. For example, the blastocyst, the morula contain totipotent cells, and blastocyst, when it comes, these cells are specialized cells. So they're going to make the placenta we call extra embryonic tissues. Now, if you take cells from the embryo, these cells cannot make again the placental cells. So that's a pluripotent. And multipotent stem cell we can find from adults different places. Example, bone marrow and some organs, skins, like these places we can find the stem cells. But these stem cells usually can give rise to only one kind. For example, heart stem cell, they can make heart muscles. The brain stem cell, they can make nerves. Respiratory cells, then they can make maybe a alveoli like that. They have only very limited when we use adult stem cells. Now in this question, why we cannot use the cardiac muscle stem cells to make the cornea? Now, the main point is when a stem cell differentiate, then it does one thing. What is main thing it does? The whole genes switch on, the group of genes, and rest of the genes switch off. So this is the main thing going to happen when this has all the genes, group of genes switch off and group of genes switch on. Once they switch off, they never switch on again. That's a permanent switch off the genes. So we cannot activate this gene again, and only active genes, they can only make the heart tissue. So the cornea, the cells of the cornea cannot make, because these genes no longer functioning, genes off. So cells are not totipotent, which means that certain genes have already been turned on or off. As a result, these cells cannot develop into corneal cells. So we have to say the heart muscle cells are not totipotent. So then the genes are switched off. So it cannot make cells of the cornea. Explain why chemicals from the eye are needed to produce corneal cells from a suitable source of a stem cell. Now this stem cell, the main thing you need to remember, differentiate into a other type of cell, for example, the sperm cell, example. How this happen? At the moment, our understanding is very limited, but we always confine to one thing: there should be a chemical. This chemical switch on a group of genes, and then rest of the gene switch off. When they switch on this gene, they produce proteins. This protein permanently modify the cell. For example, let's take a very basic things like chemical switch on certain genes and it produce protein. These protein switch off other genes, and this protein make the cells into sperm cell. So this concept is very important, and I have explained you many times, and I ask you to remember these 5, 6, 7 points and the 4 marks for these questions. Most of the times when the question is asked, many students, they cannot write even though they know this 5, 6 marking scheme points, because the question is confused. Now, for example, if you see this in the exam, why chemical from the eye are needed to produce corneal cells from a suitable source of stem cell? This doesn't show any hint that you have to write this differential gene expression theory. So you have to practice, and you have to keep in mind differential gene expression theory usually carries several marks, and you start somewhere to write answer. Always look, okay, 4, 5 marks. Is it differential gene expression? Double check, and then you can write differential gene expression, because it is asked indirectly. So these are the points. There should be chemical stimulus, and some of the genes switched off and some genes switched on. Switch on gene transcribe into messenger RNA and then translate into protein. So epigenetic modification is another concept coming from outside to make the differential gene expression. And protein synthesis take place. Protein permanently modify the cells to become specialized. So these 7 points you need to organize into a proper way to write the answer. Next one, the society's opinion about stem cell. Human embryos are another source of stem cell used to produce in medical therapies. Describe the decision that society has to make about the use of these embryonic stem cell. Decision of the society. Again, as we mentioned, most of the times ethical issues are there, and society is accepting, rest of the society accepting these things. So ethical issues are use of embryos and other things. So people need stem cells for organ transplantation and many diseases to treat, and society is again divided into 2, and they have 2 different opinions use of stem cells. Okay, now one can argue embryonic stem cells can be used to wider range of therapies, and people need these organs. The other group use of embryonic stem cell destroy embryo. So they also coming up with this idea. So I try to write the answer for this. Embryonic stem cells have the unique ability to develop into any cell type, making them valuable for various medical treatments such as myocardial infarction and some diseases like brain diseases, for example, Parkinson's, and the diabetes. When the insulin secreting cells are no longer usable, then we can use these cells. However, the origin of these stem cells must be carefully managed. There are significant ethical concerns since using embryonic stem cells involves the destruction of embryo. The killing of the embryo is an ethical issue. Additionally, it is important for research institutions to operate under strict regulation or licensing to ensure responsible practice. So in my notes, I gave you ethical issues and also role of the regulatory authority. So regulatory authority, they will oversee this use of stem cells, and the societies has this two kind of opinion. Okay, next question. Salmon are fish that produce gametes by meiosis. Crossing over during meiosis produce new combinations of alleles in the gametes. A male salmon has a genotype A, capital A, simple A, for one gene and capital B, simple B for another gene. Both of these genes are found on the same chromosome and are linked. So these are the linked gene. Complete the table to show the percentage of allele combination seen in the egg cell of this female. Now, percentage of allele combination in egg cells. So A and B, it's a higher 49.5. When it comes to a capital A, simple B is 0.5. So now if we want to fill this rest of the values, we should understand what exactly the question is saying to us. For example, same chromosome are linked, so there's a linked gene. So when we want to show the linked gene, again it's like this. Now I give you this example that you can understand well. Now let's say in a class, some of them are friends. They always sit in the same place. That means they are sit together. So let's say this is a chemistry class, and these two students, their friends, they sit together. But these two students, they are sitting distal to each other. So when we go and see another class, for example, physics or biology class, we can see these two students in biology class or physics class, they also, again, they sit together. They are always sitting together. Linked gene are like that. So when they are going to chromosomes, they always go to the same chromosome. But if they are not linked or they are distal to each other in the chromosome, then there's a chance of separation during the meiosis. Now, I can show you capital A and capital B like this. They are in the same chromosome and very close to each other. In that case, recessive one, we can see in front of capital A because they are homologous chromosome, and simple B is here. Now you see capital A and capital B, they are going to always 49.5%. Then simple A, simple B, they are also very similar to capital A, capital B. They are just very close to each other, the same length. Then we make assumption, then it's again same 49.5%. So then capital A and simple B, 0.5%. So this is 0.5 and 0.5%. So that's a percentage. Right now, the diagram shows possible arrangement of these genes on one of the chromosomes of the female salmon. So capital A, capital B, they are quite distal, arrangement 1, and arrangement 2, capital A and capital B is very close to each other, meaning we can consider arrangement B is a linked chain. State and justify which arrangement is most likely to produce the result shown in the table. So arrangement 2, because genes are located close together, therefore more likely to be passed down together during inheritance. So next is a simple calculation. A female salmon can produce up to 1,600 eggs per year for up to 11 years. Calculate the maximum number of egg cells that this female salmon could produce contain the allele containing AB in. So AB frequency is 0.495, and you multiply this by 1,600 and multiply by again to get the number 11 years. So this is the number, and then you multiply by 11 years to get the total number. Okay, next, homologous chromosomes pair up during meiosis. The diagram shows the loci of these genes on two pairs of homologous chromosomes. Draw diagram to show crossover for chromosome 1 during meiosis. So chromosome 1 during meiosis. So usually we have to show a diagram, two homologous chromosomes with crossover occurring between two chromatids. This represents the exchange between non-sister chromatid. That means this one, the part of this one is going here, and this one is going to this place. Same thing can show here. The part of this is going here, and this one is going here. Explain how crossing over and independent assortment can produce gametes with new combination of these alleles shown on diagram of two pairs of chromosomes. So in meiosis, we only focus only two things. Prophase 1, crossing over take place. Metaphase 1, random assortment or independent assortment take place. And these two bring the genetic variations. Very important to bring the genetic variations. So crossing over occurs when chromatid create new combination of allele. For instance, you might see combination of capital A simple B, simple A, capital B because of exchange of the DNA. So independent assortment, we can see the pair of chromosomes arranged at the equator, and this is completely random process. That means the chromosomes on this side and this side, there is no special arrangement. So when you have a 23 from mother, 23 from father, this could be father, this could be mother, this could be father, this could be mother, this could be father, this could be mother. So likewise, they randomly organize to make the best combinations. So independent assortment means that various combination of chromosomes like 1 and 2 are created. This results in allele for capital A and capital B potentially being found in the same gamete as either capital C or simple C. Explain how crossing over may differ in sex chromosomes. Now, sex chromosome, X chromosome is a really large one. Y chromosome is a really small one. So then crossing over is not very efficient because they are proportionally different. So when you take the chromosome like this size and the second chromosome like this, so then the overlapping like things are very efficient. But X is like this and Y is like this. So there is a very inefficient in crossing over. There's a only small amount of DNA can be exchanged, but we usually coming up with the idea crossing over can't happen between certain part of X and Y chromosome because they aren't homologous. The homologous mean they should be similar in length and most of the things between the homologous chromosomes, because Y chromosome has small number of genes. X chromosome has many number of genes. The Y chromosome is shorter, and there are specific allele present on X chromosome that aren't found on the Y chromosome. The last question, monozygotic twins develop from a single fertile egg cells. Dizygotic twins develop from a two separate fertile egg cells. This is identical twins and non-identical twins. Describe the role of cell division in the production of monozygotic twins from a single fertilized egg. So you can see after fertilization, zygote divide into two cells, and if the two cells separate and then they share the same placenta and they develop into identical twins. On the other hand, this question is asking what is the role of cell division. So zygote undergoes mitotic divisions, resulting in the formation of genetically identical cell. So that's genetically identical cells and mitosis. Mitosis and produce genetically identical cells. Body mass in human is a polygenic characteristics. The best definition of polygenic characteristic is one that involve multiple allele at a single locus, multiple allele at multiple loci. So polygenic, so let's say these are the chromosome, one here, one here, one here, one, like that. And these alleles finally decide the body mass. Next one, the body mass of group monozygotic twin was studied in 19 years. The correlation between the body mass of each set of twins are calculated. This was repeated for group of dizygotic twins. If a set of twins had the same body mass, they would have the correlation of 100%. So we don't see 100%. And from the 19 up to the 19 years, even this one is identical twins, monozygotic. That's a monozygotic, and this one is a dizygotic, and they are non-identical twins. So non-identical twins, we can see correlation is decreasing with the times because they are genetically different, and this is genetically same and they remain same. Comment on the effect of genetic and environmental factors in determining the body mass of the twins shown in the graph. Right here, they actually shows the questions twins, and you have to give 4 marks. You have to get 4 marks. You have to comment the genetic and the environmental factors determining the body mass of the twins showing in the graph. Now, which one you focus more, monozygotic twins or dizygotic twins? Right now, you have to decide what is the most important thing about this study. Then dizygotic mean they are genetically different. If they are genetically different, then it is obvious the body mass has not, there is no correlation. So it is decreasing with the time, because when they grow up, genetically different, many genetic factors affect the body mass. But when you do monozygotic twins, we don't see changes. So they remain same. Correlation is up to 90%. But the slight variation can happen. It is not 100%. So we have to be careful. So this 90 to this 10% around, we have to come up with the idea this could be environmental, because 90% is genetics. So when we're going to describe those things, eventually we focus more on the monozygotic twins. And as usual, comment question, we use a DEX format. What is a DEX format? Describe the overall result, and when possible, if the mechanisms are there, explain the result. And third one, compare and contrast, and statistical analysis. So we use these things, but for these questions, There is no statistical test done showing this standard deviation or like things, and we can generally come up monozygotic twins show higher correlation than dizygotic twins at ages. So we compare, right? So that's a C part, the compare and contrast. We're going to say there is a higher correlation of monozygotic twins, right? Show higher correlation than dizygotic. Dizygotic twins show less correlation. Therefore, genetic factors have a greater effect. So this is E now. Explanation. Genetic factors have greater effect because monozygotic twins have identical alleles. So as I told you, 90% is the genetics because they have identical alleles, and monozygotic twins show less than 100% correlation. Therefore, environmental factors affect body mass. So this 10%, we can come up with some environmental factors. So we more focus on the monozygotic twins. Right, this is the answer. Monozygotic twins have stronger correlation in traits compared to dizygotic twins at every age, indicating that genetic influence play a more significant role. This is because monozygotic twins have same allele, and it is not 100% because of the genetic factors. Next is the epigenetics. Epigenetics is change of the gene expression without changing the DNA sequence. So in theory, I also showed this picture. If the identical twins, when they grow into adult, when they have a different environmental conditions, so this person ended up in a different place, hospital, and this person still young and active, and he has no bald hair and is healthy, and this is due to the environmental factors, because environmental factors can affect gene expressions. So we learn this theoretically. It could happen in many ways, and at least two methods we learn, and this is again one, identical twins. They can end up in different situations if the environmental factors play a role. Epigenetic changes can cause monozygotic twins to have different body mass. Explain how epigenetic changes can cause differences in characteristic. Here's another picture. When the grandmother smoke, this affect to the daughter developing a fetus, and also the cells of the next generation, grandchildren, because the developing a fetus here also develop the gametes or ovum, and then the smoking like things can affect both the daughter and granddaughter because of the grandmother's habit. That's the chemicals coming from cigarettes can change gene expression. That's again the explanation for epigenetics. So mechanism is, so if we see this is a gene, and this gene, you can see the histones wrap around, and this area is a gene. So this gene, methyl group is added. After methyl group is added, then it compressed. When it compressed, it is not accessible for transcription. mRNA cannot produce because it's no longer accessible. So that's called switch off. Methyl group adding to the cytosine usually goes switch off. Then the next one is histone modification. So here you can see methyl group, phosphate group, and acetyl group can be added to the histone. When it added to histone can again rearrange, and then they can again show up these genes to transcribe, then the messenger RNA produce. For example, now this gene is available, we call it switch on now. So these are switch off. Now these three genes here, 1, 2, 3, they are switched on. So now this is what we are telling There's no sequence change, but expression can be changed. This causes epigenetic modification. Histone modification and DNA methylation play a crucial role in gene regulation. They can either turn genes on or off. This is in turn influence enzyme production and metabolism. So then the whole metabolic reactions can be changed if this protein come into the action. So this is the end of this webinar today. I think it's very important that you write all the answers and try to study this part. And if you like this video, you can subscribe to my channel. Okay, thank you very much.